Hi everyone, it's me Nirajan Pokhrel. At first, I would like to thank my supervisors, Dr. Christy and Dr. Christina. Today, we will discuss about the soil nematodes in Lantana camara, infested environmental and agriculture site. In this presentation, we will discuss some of the outlines like introduction, limitation, aim and objectives, research questions, research methodology, research progress, expected outcomes, conclusion, and references. Introduction. Lantana camara, originating from the Americas, has become a global invasive species threatening ecosystems and agriculture worldwide. Lantana outcompetes native plants, disturbs habitats, and inhibits neighboring plants' growth through allopathy. Lantana forms dense patches, obstructing livestock and poisoning animals, leading to significant economic losses, especially in regions like Queensland. Various methods such as chemical, mechanical, and biological approaches are used for lantana management tailored to specific conditions and locations. Nematodes. Nematodes are diverse organisms found in various environments with significant populations residing in soil. Soil nematodes pose threats to crops by damaging cells, transmitting plant viruses and bacteria, affecting crops such as tomatoes, potatoes, peppers, and lettuce. Populations of nematodes serve as indicators of soil health, reflecting management practices and human impacts on the environment. Nematodes play a crucial role in soil biodiversity by consuming bacteria and fungi, influencing decomposition rates and nutrient cycling, particularly nitrogen availability. There are some of the limitations while doing this research project. Some of them are limited sample size, potential sampling bias, difficulty in lantana identification, narrow consideration of environmental variables like nutrient availability, pH levels, and historical land management practices. Aims and objectives: to explore soil nematodes population in southeast Queensland, to assess impact of lantana invasions on nematodes communities, to compare diversity in sites with and without lantana, to analyze variations in nematodes communities due to lantana invasions, to inform targeted management strategies for lantana control and soil health promotion. There are two major research questions, which will be discussed in this presentation or in thesis project, which are: what is the impact of lantana invasion on soil nematodes communities in agriculture and environmental sites near Toowoomba? How does this impact vary between areas with lantana presence and absence? Background information. Invas- Invasive species pose significant threats to ecosystems worldwide. They disturb native biodiversity and alter ecological processes. Lantana camara is particularly troublesome due to its rapid spread and adverse impact on natural environments and agricultural landscapes. The research explores how Lantana camara affects soil nematodes communities in southeast Queensland. It investigates the impact of lantana invasion on nematode abundance and richness across agricultural and environmental sites near Toowoomba. Despite worldwide spread recognition of detrimental effect of lantana, gaps continues in understanding its impact on soil nematodes communities populations. The research aims to address these gaps and provides insights into the ecological consequences of the lantana invasions. Research methodology: study area selection. Two agriculture and two environmental sites will be selected near Toowoomba. Sampling designs: randomly 3 plots per site will be selected 100 meters apart. Soil samples will be collected from areas with and without lantana. Total of 24 soil samples will be collected. Soil collection: standardized equipment will be used, GPS coordinates and environmental parameters will be recorded. Nematode extraction. Soil will be processed at room temperature. Roots will be cut. Soil will be spread on trays, and 150 gram will be weighted for extraction. Additional 100 gram will be weighted for soil dry weight. Whitehead tray methods will be used for extraction. Data analysis. Nematodes abundance and richness metrics will be calculated. Diversity between sites and Lantana presence will be compared. Conclusion. Lantana's impact on nematodes communities will be analyzed. Implications for management strategies and soil health will be discussed. Limitation will be highlighted and suggested for future research. Research progress. Work done. Soil samples collected from agriculture and environmental sites near Tumba. 24 samples collected under and away from Lantana camara populations. 3 samples collected from Lantana rhizosphere pair with 3 samples 5 meters away. Samples taken up to a depth of 100 cm within 20 times 20 cm quadrant. Collected soils spreads on plates, root cords and portion weighted. 150 gram allocated for nematode extraction, 100 gram for soil dry weight determination. Soil samples dried for 48 hours at 100 degrees Celsius for accurate dry weight estimation. Whitehead tray methods employed for nematode extraction. After 48 hours, water containing nematodes collected through filtration. Nematodes counted and identified under the compound microscope at 40 times magnifications. Work identified. Analyze data to calculate abundance and richness metrics for each soil sample after nematodes population extraction and identification. Compare nematodes diversity between sites, including those with and without lantana presence, to assess lantana's influence on soil nematodes communities. Interpret findings from data analysis using R statistical software and MS Excel to understand the overall impact of lantana on soil nematodes populations. Discuss implications for lantana management strategies and soil health conservation based on the interpreted findings. Expected outcomes. There are some of the expected outcomes. As some of them are: understand how lantana invasion affects soil nematode communities. Compare nematode numbers and types in areas with and without lantana in both agriculture and environmental sites. Explore why nematodes populations differ in places with and without lantana infestations. Analyze how nematodes diversity relates to factors like moisture levels and the types of plants present. Use findings to develop better ways to control lantana while promoting soil health and biodiversity. Conclusion. This research project focuses on the impact of lantana invasions on soil nematodes communities in agriculture and environmental sites near Tumba, variations between areas with lantana presence and absence. Overall, this research is expected to inform targeted management strategies for lantana control and soil health promotion. And these are the references which are used. Thank you.